Thank you, and thanks for inviting me, and thank you for coming and listening. So I will talk today about sort of a joint or two projects by Tuba and Od. Tuba was a PhD student who already finished, and Od as a postdoc in my lab. And the sort of main questions we're interested in date back to my time as a psychology student, and to this distinction, which was actually coined many times, but about the possibility to control behaviors with two main types of systems. So this is an illustration after Kahneman idea of system 1 and system 2, where we can control behavior using a competition between two very distinct forms of control, where we have one automatic, very fast, using heuristics, the system 1-1, the system 1-1 is good, and system 2 that is much slower and deliberate and faster, to slow and able to perform plans, also very flexible, but very labor intensive or difficult to maintain. Okay, so and this distinction maps very nicely onto a distinction that people have used for many years in behavioral psychology about two types of behaviors: the habit type of behavior, which again is automatic, takes a long time to learn, but once it's done, it runs its very stereotype, and it is unaware of the outcome. So something, you see something, you just go ahead and do it, not thinking of whether or not you want the result of this behavior. Whereas on the other hand, we have the goal-directed behavior, which, as its name implied, is really geared towards achieving some sort of goal. So this enables planning, obviously. It's very flexible. You can use it. If you have different goals, you can change your behavior accordingly. It's very adaptive. You can perform planning, and it's sensitive to the outcome by the very definition. And we know sort of that the habit control is implemented in the basal ganglia. And we know that the more deliberate, goal-directed behavior is ultimately controlled by various projections from the prefrontal cortex. And this again maps to another distinction. Sorry. Ooh. Okay. Maps into another distinction between model-based and model-free behavior. And the idea of the habitual or basal ganglia type model-free behavior is that it's implemented by plasticity rules of the basal ganglia, specifically of corticostriatal connections. And those corticostriatal connections are those who provide the system with information, sensory information about the world that would come from the cortex, and ultimately would go to actions or to correct behavior or to the habitual behavior, and the plasticity rules that would cause this correct mapping from sensory information to behavior are molded by dopamine, and the dopamine informs the system about some form of knowledge about reward, and this would allow, and dopamine indeed controls plasticity in the corticostriatal circuit, and this would allow the system at the end, without any knowledge of what's going to happen, just by virtue of dopamine coming there at the right time, at the end to just choose one of the possible actions, sort of in a stereotyped manner. So that's what we know about the habitual model-free system. However, in the model-based system, in the goal-directed system, The idea is that alternatives are elaborated first in sort of an internal model of the world, of the connections in the world. And according to whatever our goal is, the person or the animal would travel mentally inside this internal model, and the internal model again could be a proper map, or it can be anything else, and figure out what the outcomes of each route would be, and then choose one according to that. So the idea of an internal model in that sense, in the action selection sense, was first coined by Tolman in mid-1900s, and he called that internal model, and again, that's an internal model in any sense you would like, as a cognitive map. So our hypothesis is that the internal model for behavior, or the cognitive map, represents the parameters that are important for the task in reward relevant dimensions, because you can imagine that you can represent the world or the same type of sensor information in different types of ways, and there would be those ways that would be more relevant or more informative to achieving a goal than other ones. And our hypothesis, furthermore, is that this representation, this change in representation, is governed by the same plasticity rules, dopamine-dependent plasticity rules, that's good for the habit system. So we have one projection, it tells the world about upcoming rewards, and therefore it may teach us about correct actions in this way. Maybe it also teaches us about the correct way to represent the world, also for goal-directed behavior. So we're going to check that. And then indeed, dopamine projects to all of those places that are relevant for both types of control. So that's a scheme that I shamelessly stole today from Google. But dopamine comes, of course, to the striatal system for the habit control, also to the prefrontal cortex, various parts for the goal-directed control. But I would like to show you that it also comes to other places, among them to the hippocampus. So our idea is that in the hippocampus, which we claim would be the internal model for goal-directed behavior, dopamine shapes the representation such that it will be optimal for behavior. So the idea of a hippocampus, of course, as a cognitive map is an old idea, and the idea there, which stems from O'Keefe and Nidell from '78, they wrote a book, The Hippocampus as a Cognitive Map, and there they use the term cognitive map as a physical type of spatial map. We would like to claim that the idea is more general for any type of behavior. So a map, so the hippocampus could be an internal model for whatever task you may want, and if the task is spatial because you're a rat navigating through a maze, then that map would be spatial, but not necessarily. So we want to ask, does reward affect the type of representation in the cognitive map in the hippocampus? And assuming it does, we would want to ask how. So how do we study what is the representation in the hippocampus? We will use two very prominent properties of place cells in the hippocampus. The hippocampus contains place cells, so those are neurons that fire specifically in one location in the space. And those neurons are very stable in their representation, so this is what we see here from two papers from the Moser lab. We can see over here is that if you take a cell, record its activity in a certain arena, remove that cell, and then reintroduce it to the same arena, the cell will fire very similarly. So its place, it will represent the same point in space. However, if you take now a cell and move it through different environments, as you can see there on the left, the neurons will remap or change its spatial representation according to the different environments. And the third very important point is that if you now take a neuron and take a rat and reintroduce it to a different environment, but that is sufficiently similar to an environment that the cells fired in, the presentation would be very similar. So we can use this balance between stability and remapping to try and figure out, try and dissect what it is that, what the representation is for these neurons in the hippocampus, and whether that changes as a function of relevance to a task, to goal achievement, and or as a function of dopamine. So this is what we'll do here. So this is just schematic illustration of what I mean. So if we think of this schematic rat over there in this square black arena, and we record a cell, and it represents this point in space over there. And we now reintroduce the rat to an arena of the exact same dimensions, just a different color, and look at the firing of the place cell. We will see that this schematic place cell did not change its representation, meaning it doesn't care about color. And similarly, it wouldn't care maybe about the length of the arena, but if it would remap, if we moved the animal to a similar arena but of a different width, then we would conclude that actually the metric for space at least for this particular cell would be width. So all of the other dimensions of space are not relevant for representation of this particular cell, and then we can record a large number of cells and try to understand what the representation of the entire population is. So this is, yeah.
The picture that I have in mind is that remapping is a very global phenomenon. It's not that one cell cares about length and another one cares about something else, but it's all the cells remap together. Are you challenging this?
Not at all. Okay, so the first thing we wanted to do is to try and see whether we can sort of entrain the whole population of the hippocampus from encoding space in one set of coordinates to a completely different set of coordinates, in this case, olfactory coordinates. So this is what Tuba did here. She took rats, she introduced them to this ring type of maze in complete darkness. She would always put them in one particular location in space, and the rats had to run around and look for a drop of water. These are thirsty rats, and there are 16 holes in this maze, and through 4 of them, 4 different odors are continuously pumped and sucked out. And so what you would do here is introduce the animal, give it 2 minutes to run, remove it, rotate the odors, reintroduce the animal, and do the same thing for 10 trials a day, 10 2-minute trials a day, where the reward could follow 2 different rules. So the reward for one set of animals, for one group of animals, followed an olfactory rule. So this is what's depicted here. No matter how much you rotate the odors, the reward will always be, 12 degrees clockwise from this green odor. Okay, and for a different group of animals, they would do exactly the same thing in darkness, rotate the odors, but this time the reward would follow a spatial rule. So no matter how much you rotate the odors, the reward would always be in this location.
Genela, do they have any other spatial orientation except for the odors? Are there any other cues that they can use?
I would want to say no, but that would be a lie. The experimenter always enters in the same location. I'm pretty sure there is buzz from a computer that's outside, but it's still there. And very, very importantly, the animal always enters in the same point of space. So in principle, it can count steps. So the animals, both groups of animals, learn how to do this, and they learn it pretty slowly, which is lucky for us actually, because it gives us the opportunity to see what happens during this learning. So it takes them about 10 days to become good, and we continue recording way over those 10 days. So what do the cells do? So in principle, we can see two types of cells. They're exemplified here. This is a cell that's recorded with a rat that ran in the olfactory rule type of task. So how would I use this? I won't use this. Okay. So this is the arena. Thank you. This is the arena. What's color coded here is the firing rate of one particular cell. So this cell in this case prefers this location in space. And now we'll rotate the odors, and you will see that the place field of the cell rotates together with the odors. Specifically, it's pretty close to this red odor over here. Okay, so that's one type of cell. And then we can look at the relation between the orientation of the maze and the orientation of the firing angle, the best firing angle of this neuron, and you can see it's really nicely correlated. So that was the cell we saw before, but we also have other types of cells, and this is a cell that was recorded with animals that ran in the spatial rule. So same thing. This cell prefers this. In this orientation, prefers this location in the arena, and as we rotate the maze, it continues preferring the same location in the arena. So again, if we correlate maze orientation with the cell orientation, we get nothing. Okay, so those are two types of cells, two extreme types of cells that we can see, and if we now look at all of those correlations between the maze orientation and the cell orientation, in the two groups of cells, so the histograms are depicted here. The green cells, the green bars, are from cells recorded in the olfactory rule type of condition, and the black bars are recorded in the spatial rule. And you will see that those distributions are clearly really very different, where in the olfactory rule you can see a concentration in very high correlation and perhaps some bimodality. We also have some very low correlations, and in the spatial rules, most cells are concentrated over here. So in order to quantify that a little better, we wanted to try to really see what those neurons are encoded, really not just in terms of correlation to the maze orientation, and basically we wanted to see how closely the cells followed the spatial rule, so how similar were they to this type of line, versus how closely they followed the odor orientation, how close were they to here. So we just divided an index, pitting the two one against the other. So in this index, if you get an odor index of 1, you would be a perfectly odor coding cell, and if you get an index of -1, you would be a perfectly spatial coding cell. And since we recorded the neurons, the animals during the full behavior, we can see how that evolves through time. So you can see that both groups of animals start with a pretty spatial or negative values for quite a long time. Okay, so the white marks here are the animals that use the spatial rule, and the green marks are animals that ran in the olfactory rule. So they start pretty spatial, which is good. I mean, someone beat us to the Nobel Prize, but I mean, it's Hippocampus. So that makes a lot of sense. But with time, you can really see a divergence between the two groups, where one group starts encoding space in olfactory coordinates and does so very well, and the other group becomes better and better at encoding space and spatial coordinates.
According to the training, that's what they did, because this is what we were trained. But can one cell move from one to the other?
So
we didn't test it when you have two groups, you can test the same cell, but did you test it on both?
Can you wait?
Okay.
Just a clarification, so the reward itself can serve as a spatial key, right?
That is true.
For the spatial.
That is true. Which is the real problem for us. So what we try to do is try to see whether we can find enough neurons where you can see the correct mapping before the animal actually encounters the reward. Because you could imagine two things. One is you have a very, very strong cue. In one sense, it's correlated to odors, in the other sense, it's correlated to the room, and that may reset the system. Or you could say, no, it's a long-term plasticity, and the animal already learned how to use it without using the actual reward. I don't have a good answer for that. The reason I don't have a good answer for that is that at this time, the animals are really, really good. So they get to the reward very quickly. And then I don't have enough, I mean, I do have examples of cells that, of neurons that encode space in these coordinates before they get to the reward, but they're not that many. So I don't know the answer.
But they learn quite slowly, so you have time in between, before they are so good, you see. So
Yeah, but when they get, so around here, day 10, when they actually diverge enough so I can tell you that those neurons are encoding space in those coordinates, they're already very fast. Okay, so we can see that hippocampal map is really shaped by the reward or the type of information that's needed for reward obtainment. And so the next question is the question of mechanism, of how is this done. So for that, Odd here used a slightly different task, and this task is a bit easier for the rats, which makes it faster, which is fun for us. So in this type of experiment, we don't have olfactory cues anymore, so everything is visual. It's done in the light, or at least in dim light. And so the animals are introduced into this type of arena, which is basically a big bowl that has 100 tiny wells in it, and one of those wells would be baited with, again, a drop of water. And now the animals can navigate according to two sets of cues. One set would be located inside the maze and would be proximal to the maze and would move together with the maze. The other set of cues are outside or distal cues. And then, again, the animal is taken in and out, the maze is rotated, and we have two different types of rules again. A distal rule where the reward is always in the same place in the coordinates set by the distal cues, and a proximal rule where the reward, sorry, this is the orange one, the reward is located in a constant position in proximal, in the space defined by the proximal cues. And so the experiment is the following. She trains the rat on one rule, in this case in the distal rule, for 5 days, 4 to 6 days, until they become pretty good at it, and then she switches them to the other type of rule. And while she switches them, since our hypothesis is that this reward information is conveyed by by dopamine, and that dopamine is responsible for the plasticity that causes this type of shift, then after the shift, she infuses, she micro-infuses a D1 receptor blocker, specifically to area DA1 of the hippocampus, which is where we recorded the neurons. So she infuses this antagonist on every single one of these 5 days in which the animals are supposed to learn the new rule, after supposedly their map already reoriented according to the first rule. And just to make sure that there's not something that's specific for one rule over the other, we also do it in the opposite order. So this is what the animals do. Okay, so what you see here is the animals that started with the distal rule, or the animals that started with the proximal rules, two groups of animals according to what we will infuse them later, and of course they learn it pretty quickly, within 5 days, so they're pretty good at that. And then we switch them to the second rule, which they don't know, of course, and we start infusing the dopamine receptor blocker into the hippocampus. So the first thing we want to make sure is that those animals to whom we infuse the blocker are still motivated enough, and we didn't change anything very drastic in their reward system. So we want, or in their motor system. So we want to know that they know where they're going, and for that, since they don't know, we changed the rule. What they should do is run to the well where the reward should have been given the previous rule. So we compare that, and they're exactly the same. The two groups. So the blue ones are the block group. They're exactly the same. So they can run, and they know, or they think they know where they're going, only they're wrong, right? They have to learn a new rule now. So what happens for the first rule? So the control animals are pretty good at learning the second rule as well. No surprise with that. The dopamine receptor blocks animals are very bad at learning the new rule. They improve very slowly. They never become as good as the control animals, and they're dumb. Okay, but if you want to try and figure out what do we mean by dumb, you can not only measure the time it takes them to reach the reward, but actually what they do in order to try to get to the reward. And in this example, you can see the trajectories of two animals, of a control animal and of a dopamine receptor blocked animal. So the black lines, which I hope you can see, are the trajectories. The green stars is where the new reward is, and the orange stars is where the reward would have been had we not changed the rule. And what you can see is that while the control rat initially, of course, looks for the reward in the old well, can't find it, and then starts searching and gets to the reward, by the last day definitely, but also in the middle day, he already does sort of a direct pathway towards the reward. Whereas this blocked animal, which also finds the reward at the end, would always go through the previously learned rule first. So it sort of perseverates. It can't understand that the previous rule actually doesn't work anymore. So we quantify that. So this is the fraction of trials within days in both rules that the animal went to the old wrong quadrant, or wrong well actually, first, before going to the first one. And the control animals are very fast at not doing this anymore, but the dopamine blocked rat continues searching, first of all according to the old rule, then they get disappointed, and then they go and start looking for something else. So this is in terms of the trajectory itself, where did they go first, and this is in terms of how much time they spent in each of the quadrants, and basically you can see the same thing. Yeah.
So for me it looks like it could be one of two things. One is that there's cognitive inflexibility, or one is that their learning is impaired, period. And that had you done the antagonist for the initial rule learning, they also wouldn't have acquired that, and it's not only the transition between.
So we did antagonist for the initial learning. They are slower to learn,
But not
this
to this
bad.
extent.
They are slower to learn though. Definitely there's not that. But I would claim actually that I'm not sure if this is inflexibility per se. Flexibility implies that you have knowledge of the two options, and you can't really choose between them according to context, or according to motivation, or according to something. I'm not sure that the new one actually exists. But since we don't do the back and forth, I don't know. Okay, so that's what's happening in terms of behavior, what's happening to the neurons. Was that enough? No. I'm not out of time yet, am I?
No.
Okay. So what what's going on with the neurons at this time. So what we see here are 10 trials of one type of cell. This cell was recorded in the distal condition, so you can see the reward is always in the same place, and the place field of the cell in all 10 trials are also in the same place. So we can superimpose the trajectories of all these 10 trials together and the spikes emitted by this same cell on all 10 trials, and we can see that we can still get a very nice cluster of spikes in this location, or we can translate it to a race map, and we'll see that this neuron is very directional in the distal coordinates of space. So that's this neuron. However, if we now reorient sort of our maze in the proximal coordinate system and superimpose the trajectories and the spike together, the cell loses its directionality, it loses its place field. You can see it here, and you can also quantify this. So the white marks are the angles of the cell in the proximal coordinate space, and the dark ones are the coordinates of the best angles of the cell in the distal coordinate space, and you can see that it's very directional in the distal coordinate space and completely uniform, or almost completely uniform in the proximal space. And we can test that by computing the Rayleigh score and by testing its significance.
Again, just a clarification question. So all these are 10 different trials?
10 different 3-minute trials.
And you compute a map from how many spikes?
Those ones?
Yeah.
So in total for all, I think she put a threshold of minimum 30 spikes per trial, and at least 3 passes through each point in space.
This is sufficient for generating these maps?
This is sufficient for looking at those maps. We tried different thresholds. This is obviously based on many more spikes, so it's consistent. I mean, if you have a single spike, then obviously you will get a really, really nice place field. So you need to have enough spikes for that, enough statistics. So we would term this type of cell a distal cell. This is a different cell, also recorded in the distal configuration. So the reward is still in the same place, but this place field of the cell clearly rotates, and I'm sorry, but I lost here the proximal cues to show you how this actually rotates together with the proximal cues, but you can see that over here. So these are all of these trials superimposed on each other, and the cell loses its place field, it loses its directionality, whereas if we align them according to the proximal coordinate frame, we can see that the cell is indeed proximal, and you can also see it over here in the white marks. So we would term this type of cell a proximal cell, and then we would have cells which look like they have clear place fields in each one of the trials, except we can't figure out which space they encode, if at all, and that's sort of depicted here. So they don't encode anything very systematically clear to us, at least not in these two coordinate frames, so we would call that an unidentified cell. And so we can look at all the cells we have together. So this is all the cells we recorded in all conditions, and plot them according to the results of this test, so the Rayleigh Z-score, and you can clearly see that there are 3 clusters here. There are cells that are very nicely clustered on the distal side. very nicely clustered on the proximal side, and a number, quite a large number of cells that are unidentified cells. So now again, we want to see how the population itself, and the entire population encodes space, and whether that changes through learning and through relearning, and what dopamine does there. So this is what we have here. So those are cells from animals that were recorded in the distal first type of experiment. So we start with the distal configuration, and then we switch to the proximal one. So those are the proportion of cells, of distal cells, proximal cells, and unidentified cells throughout learning. And as we saw also in the previous experiment, most of the cells start by being distal in the first place. They start by being proper spatial kind of cells, and this proportion increases mainly at the expense of unidentified cells, the proportion of which decreases. So it might be that those are recruited to encode space in those coordinates, which we can't see. Okay, so this is what happens before the infusion and before the shift, and then we perform the set shift and the infusion of the dopamine blocker, and this is what we get. So the neurons start sort of similar to here throughout the 10 trials, and they start reducing the representation of space in distal coordinates, and increasing space representation of space in proximal coordinates, and we completely at some point lose the unidentified cells. So it really looks like there is a recruitment from one sort of representation to the other sort of representation, and about at this point the proximal configuration actually wins over the distal configuration, and it nicely, although unstatistically, coincides with a time where the animal stops searching first in the according to the previous rule. I cannot support this claim. I will deny I ever said that. So this is what happens in the control condition. What happens when we block the dopamine projection to the hippocampus? So we lose. First of all, we start also reducing the number of, or the fraction of cells encoding space in distal coordinates. However, we do not have a recruitment of the unidentified cells towards the proximal one. It stays about the same, and the proximal one increases slightly, never wins over the distal configurations, and indeed those rats continue searching for the reward in the wrong place first.
It looks like maybe if you will go to day 6, maybe it would grow.
It could. You're welcome to come and do the experiment. Okay, so they do improve in time, but they never actually, it looks like they never, at least within those 5 days, never, or are at least slower to change the map that they have of space that they have in the hippocampus. Only it's not only that, the quality of representation is also different, and this is what I want to show you here. So those are control rats. After infusion, those are the size of the typical place field for one particular neuron. This is one particular neuron. This is very representative of the dopamine blocker cells, and their place fields are substantially larger than the place fields of the control rat. So the resolution that they can encode space in is substantially worse, even if they do learn the type of representation that they need to have in order to correctly perform their task. So this is what we have here.
Is that, so for proximal cells and distal cells, do they basically have a size difference also?
They don't have a size, or not a statistically significant size difference. No, they don't. And they all grow together in the dopamine blocker condition. Okay, so in summary, we saw that the animals learn to represent space in the coordinates that are actually relevant for their specific task, for achieving their specific goal. That is determined by motivation, so I didn't show you that, but we have experiments where we can play with animals' motivation and show that what they define as goal, and hence the representation is in the hippocampus, is actually molded by their motivation. And we show that the local dopamine in CA1 is necessary both for the behavioral and the neuronal reorientation of the map, or a change in spatial representation that we see in the hippocampus. So I know I'm almost out of time, and which is good because the next thing I want to show you is very, very preliminary and short. But the big question is, where actually does the dopamine come from? I started by giving all of this motivation about the dopaminergic, the relation between the dopamine system and reinforcement learning, and how the dopamine signal, which we know exactly how it changes in different conditions where the reward is more or less predictable in order to optimize learning, and we show how that same dopamine actually changes the plasticity in the hippocampus, except it's not really clear that there are very strong projections from the VTA to the hippocampus. There probably are such connections, but definitely a large fraction of the dopamine that gets to the hippocampus, or to the dorsal hippocampus, and specifically to the dorsal CA1, is actually co-released, or possibly, I want to be careful here, possibly co-released from fibers from the locus ceruleus that actually usually release noradrenaline in the same region. So if the effect we're seeing here is actually an effect that comes from the locus ceruleus, then it would hint that this is actually not a plasticity and learning effect, but an attention effect. So it could be that the animals learn to attend to the correct system, regardless of the dopamine and the reward. They already learned to do that. They're more attentive. This attention is perhaps enhanced or at least in some way mediated by noradrenaline, and this noradrenergic signal releases dopamine together with it. So which is it? So what we're trying to do now, and again, this is very preliminary, so I could be completely wrong. We are using TH3 animals, and we're lucky because both noradrenaline and dopamine neurons express CH, so we can use those animals and repeat our experiment by injecting inhibitory drug either to the VTA or to the locus ceruleus. And repeat the same experiment that we're doing, except we don't want to activate those dreads systemically, because we really don't want to interfere with the arousal system or with the reward system. I mean, I can tell you, if we change something substantial in the VTA or in the locus ceruleus, the animals won't learn and they won't behave. But that's not very interesting. We want to know specifically which projections to the hippocampus are important for our type of relearning of a map. And therefore, we inject our CNO locally and CA1. So we affect specifically the projection from VTA to CA1 or from LT to CA1. So this is what we see here. Those are animals learning before. And those are the three types of animals after the rule shift. So those are the control animals. They learned the rule shift pretty well. So those are days 1 to 5. Those are the VTA injected animals. They are much slower to learn. They start much worse. At the end, in this case at least, they're just as good. Those are the LC injected animals. So they start somewhere in between and they never improve. But what's interesting is what happens within a day now in these groups. The black bars are the control animals, are the regular animals. Dark bars, filled bars are the beginning of each day, and the white bars are at the end of each day. So the animals learn here, and in the VTA they are worse to learn, but they still improve between days. What we see with the LC injected animals is something completely different. So they start bad, they improve significantly within a day, they go back to being bad, they improve significantly within a day, they go back to being bad. They just either don't remember what they did the day before, can't retrieve it, haven't consolidated it, one of those things. So we don't know what the answer is. We do know that this effect is not mediated by the noradrenergic receptors, because we have done the experiment. I don't know what this means. And I think...
They have normal sleep. Those, the locus coeruleus.
The CNO washes out. So I don't know. I don't...
Consolidation might suggest.
I don't know, but we have 24 hours between each set of experiments, and the CNO washes out already. So I'm not sure we're interfering with the sleep, but I'm also not sure we're not. Okay, I think that's it. Exactly in time, 1 minute before. So just thank you to present former lab members, Odd and Tuba, who did this work, and the dread work Odd is doing in Thomas' labs in Bordeaux.